Assalamualaikum warahmatullahi wabarakatuh. Good morning, honorable judges, all of this participant conference, and also all of this organizing committee of AMRC 20 and 24. Let me introduce myself first. My name is Nur Aliya, and I'm a last year in medical student at Islamic University of Indonesia. It is a pleasure for me to present my research, especially in the basic science, about the effect of simvastatin and ascorbic acid encapsulated CHITOSAN nanoparticle on the thickness of seminiferous tubule epithelium in WISTAR rat induced by carbon tetrachloride and high-fat diet. Okay, as we know, a high-fat diet can trigger the level of oxidative stress, so it can cause a hyperlipidemia, which has been shown to contribute to male infertility if it's prolonged and not treated properly. It turns out that stress oxidative can damage the testicular tissue, especially the plasma membrane cell, which is leading to structural damage. This damage manifests as a change in the histomorphological of the seminiferous tubule, which are critical to spermatogenesis. And then it is important to explore some potential therapies to mitigate this negative effect caused by hyperlipidemia. This research approached the use of statin, especially simvastatin, as a drug primarily used to reduce the lower cholesterol level, and also then also ascorbic acid as an antioxidant, and both the combination as a drug are expected can counteract the increase of oxidative stress. But however, the bioavailability of this compound are often limited. To improve this, we have to encapsulate simvastatin and ascorbic acid into nanoparticle chitosan to enhance their effectiveness in treating hyperlipidemia-induced damage. So, the aim from this research was to evaluate the effect of combination both simvastatin and ascorbic acid in the thickness of seminiferous tubule and WISTAR that had been induced by carbon tetrachloride and high-fat diet. This was a purely experimental study using a post-test only control group design, and we also used a total of 28 male rats, which are randomly divided into 3 groups. The first group is a normal group, which is the rat were only given a standard feed without any intervention. And then the second group are given a treatment, which is the rats were given a standard feed and high-fat diet, and also a carbon tetrachloride, as much as a 2 milliliter for each rat per day for 4 weeks by a mixture of a coiled egg yolk and use a cooking oil. And then the last one, our intervention group, which is the rat were given a standard feed diet, high-fat diet, and carbon tetrachloride, and also along with a therapy, which is a combination of simvastatin and ascorbic acid induced by CHITOSAN nanoparticle as much as 0.018 milligrams per 200 grams body weight of a rat per day for 4 weeks. And then the primary outcome of this research are to measure the average of the seminiferous tubule epithelium, which was assessed after intervention period. And then we do the statistical analysis, continue by ANOVA test, and then we continue by a bond foreign post hoc test for the group comparison. The results from this each group shows that the highest average of the thickness of seminiferous tubule was a normal group, and then the second is the group intervention, which is the rats were given provide the therapy combination of simvastatin and ascorbic acid, and then the last one, the thinness, are the group were given a treatment, which is the rats were induced by high-fat diet and carbon tetrachloride. And then the statistical analysis showed that a significant difference between the group. But however, the further analysis by Bonferini post-hoc test revealed that there's no significant improvement between the group intervention or the therapeutic group compared in the group using HFD atau high-fat diet on their thickness of seminiferous tubule epithelium. In conclusion, while the combination of simvastatin and ascorbic acid encapsulated in CHITOSAN nanoparticle maybe demonstrates some potential in mitigating the negative effect of this high-fat diet with carbon tetrachloride to increase the level of oxidative stress caused by hyperlipidemia. And then even though the results suggest that it did not significantly improve the thickness of the seminiferous tubule between the intervention and the treatment group. This may indicate that while the treatment have some beneficial effect, but the further optimization of the treatment have some therapeutic formulation or additional intervention may be needed for more effect. For example, providing the therapy for a longer period of a time, or maybe replacing simvastatin with another drug choice from a statins category, or maybe we can also increase the dose simvastatin gradually. I think that's all from me. I would like to thank you for your amazing attention, and I hope this research provided valuable insight, and I look forward for any question or feedback you may have. Thank you very much.
Thank you so much for your presentation. Now you have 3 minutes for a questions and answer section.
Just wondering about you are using SIMVASTATIN and
Yes.
ASCORBIC axis.
Yes.
This is a common medicine for human, right?
Yes, actually.
So I just wondering about the WISTAR that they can have a same response to the medicine as the same as a human in the same level, because if you it's like a mouse model, right? If you want to use it for to see the result and you apply it to the human model, right?
Based on the previous research, there is a similarity between the Wistarets and the human body, because in Indonesia, in my country, is simvastatin is the most widely used to drug to reduce the level of cholesterol, and then another choices from statin, example atorvastatin, rovastatin, or many drugs in the statins categories. And this drug also have several dose. For example, 10 milligram, 20 milligram, 30 milligram, and so it can be used as needed.
According to conclusion, you said that no significant difference between the treatment group and the control group, right?
Yes.
For the control group, you feed them with the high-fat diet, which consume that they have the high lipid profile,
Yeah, correct.
right? So apart from this data, are there any other result, right? Blood test or
Okay. Okay,
whatever?
maybe in a statistical analysis, there's no significance between the rats were given the high-fat diet and therapy of simvastatin and ascorbic acid. But if we look at the histomorphological in the picture, as you can see on the screen, that the normal group, which it has a good histological, there's no disorganization between each tubule, and there's no distance in the tubule, and nothing in loose. But if we look at the intervention and the treatment group, it's revealed that a thinning of a seminiferous tubule that may indicate that the high-fat diet experience is maybe also the change the pathological, because the luminar diameter or the diameter luminar are also narrowed, and then the disruption of the connection between Sertoli cells and increase the apoptosis as a side effect of oxidative stress.
Okay. You say the increase in oxidative state can cause the increase in the thickness of the
Seminiferous tubule.
tubule.
Yes.
Okay.
Any question from our online judges?
Yes. I'm just wondering, why do you choose the seminiferous tubule as the target for monitoring? Shouldn't it more properly to state in the title is that if you want to study the male fertility function or what, and if you are going to look at the effect on the hypercholesterolemia with the treatment of the simvastatin and vitamin C nanoparticle was more effective than, I don't know, just application of the drug without the nanoparticle or what. To be honest, I'm not quite sure about your the problem, the hypothesis behind. Please clarify this first. Thank you.
Okay. Actually, this research in steatohepatitis, but if I look at the many beneficial I found that the high-fat diet can also can make many change in histomorphological and the testis, that there is beneficial for the make or the mature spermatogenesis.
My suggestion is that don't waste your rat. Not only for the tissue, but you can collect the sample and I mean the blood sample. Yeah. And you can do more experiment and
Experience,
get
yes.
more data.
Yes.
Yep.
Thank you very much.
Thank you so much.